So our next major type of macromolecule are the nucleic acids, and these you're probably really familiar with. So these include DNA here, our double helix, as well as RNA. So here we have a transfer RNA. And these are basically the molecules that store and transfer genetic information. So DNA stores the genetic information, whereas RNA transfers it. And just to sort of reiterate that function, so you know that in eukaryotes, DNA is found within the nucleus where it encodes proteins. So DNA is copied by transcription to make mRNA. These messenger RNAs or mRNAs then exit the nucleus and go into the cytoplasm where ribosomes will attach to these mRNAs, and using transfer RNAs will then synthesize proteins. So they're very important for both encoding proteins as well as being used to actually make proteins, so transferring the genetic information to the cytoplasm. So the three major types of RNA are the mRNA, the messenger RNA that codes for protein, the tRNA, the transfer RNA that brings amino acids to the ribosome, and the rRNA that's actually a structural component of this ribosome. And we'll talk more about that in the next couple of chapters. Here we do have a traditional monomer. The monomer for nucleic acids is the nucleotide. And you can see here that the nucleotide has three major parts. First, we have the nitrogenous base. The nitrogenous base is what gives the nucleotide its identity. There are 5 major nitrogenous bases that are found in DNA and or RNA. Cytosine, adenine, and guanine are found in both DNA and RNA, while thymine is only found in DNA, and uracil is only found in RNA. And these can be separated into the pyrimidines and the purines. So the pyrimidines are single ring structures, whereas the purines are double ring structures. And it's important to note that purines can hydrogen bond with pyrimidines, and we'll talk more about that in a second. So the nitrogenous bases was our first piece. The second piece is a 5-carbon sugar. In the case of DNA, this 5-carbon sugar is deoxyribose. In the case of RNA, this 5-carbon sugar is ribose. The only difference between these two sugars is what is bonded to the 2' carbon. These carbons, remember, we number the carbons starting to the right of the oxygen. This is the 1' carbon, and we use primes here just to differentiate because these carbons can be numbered as well. So the 1' carbon is what the nitrogenous base is attached to here. The 2' carbon is where either the hydroxyl group in RNA or the hydrogen in DNA in deoxyribose is attached. The 3' carbon has a hydroxyl in both sugars here. And then we have the 4' carbon and the 5' carbon. And the 5' carbon is attached to the last piece of our nucleotide. The 5' carbon is attached to this functional group, which you should recognize as a phosphate group. And remember, at physiological pH, this phosphate group has a negative charge, meaning that nucleotides are very negatively charged. Once again, just to review, the 3 pieces of the nucleotide are the nitrogenous base, the 5-carbon sugar, which is deoxyribose in DNA and ribose in RNA, and the phosphate group. Our monomer for nucleic acids is the nucleotide. So now we've talked about nucleotide structure, and I want to point out another important nucleotide. We've talked about that these nucleotides can make our polymers of RNA and DNA. However, ATP is probably the most important nucleotide in the cell. And so ATP, we've already learned, is produced by aerobic respiration in the mitochondria. And ATP is adenosine triphosphate. And essentially, it's an RNA nucleotide that has an adenine nitrogenous base, a ribose 5-carbon sugar, and then it has 3 phosphate groups attached to the 5' carbon. This is essentially the energy currency of the cell, so we can use it for energy to accomplish reactions that require energy, and we can also use it to harness energy from reactions that release energy. And that's because if you look at these 3 phosphate groups, you can see that we have these negative charges hanging out really close to one another. And you can imagine that these negative charges repel one another. Because of that, breaking the bonds between these phosphate groups releases a lot of energy, allowing us to harness that energy to perform chemical reactions. Now let's talk a little bit about how these monomers, these nucleotides, assemble into polymers. Unlike carbohydrates, nucleic acids can only assemble into linear chains. You can see here we have a phosphate group and our sugar, and then our base sticks out, and then we attach the hydroxyl group that was present on the 3' carbon attaches to the phosphate group of the next nucleotide. So a hydroxyl group that was present here on the 3' carbon attaches to the phosphate group present on the 5' carbon of the next nucleotide. If we look here, we can see that this forms what's known as a sugar phosphate backbone, because we have sugar, phosphate, sugar, phosphate, sugar, phosphate. And the bond that links nucleotides is known as a phosphodiester bond. So now we know the monomer, which is a nucleotide. We know the polymers, which are DNA and RNA. And the bond between monomers is known as a phosphodiester bond. These create the sugar phosphate backbone, and then the bases, those nitrogenous bases, stick out into space. And because these assemble in this linear manner, this means that nucleic acids actually have directionality. And that's because one end is always going to have this 5' phosphate group, so the phosphate that's attached to the 5' carbon, so we can label that the 5' end, while the other end always has a hydroxyl group that's attached to the 3' carbon, so we can label that end the 3' end. DNA and RNA polymers have directionality. They have a 5' end and a 3' end that are chemically distinct from one another. And that's going to become important in the next few chapters. So let's take a look at RNA and DNA a little more closely. Here you can see that phosphodiester bond, once again, is the linkage that links two monomers in either RNA or DNA. There's, you can see these phosphate groups that are present in both. So the 5' end always has a phosphate group, and the 3' end, if this had stopped before going on to the next nucleotide, would have had a hydroxyl group. So we have a 5' end and a 3' end. Now RNA is a single-stranded polymer, whereas DNA is a double-stranded polymer. And how these DNA strands align, they align in an anti-parallel manner. So this strand is going from 5' to 3', while the other strand you can see is shown upside down here, and that's because the 5' end is down at this end and the 3' end is at the top. So these two strands are anti-parallel, meaning that they run in different directions. And you can also see here how these two strands are linked together, and that's through hydrogen bonding of the bases. We can form base pairs between C and G nucleotides and A and T nucleotides. You can see here we have a purine and a pyrimidine that link together through hydrogen bonds. And CG base pairs are linked by 3 hydrogen bonds, whereas AT base pairs are linked by 2 hydrogen bonds. Major differences between RNA and DNA. First, RNA has ribose as its 5-carbon sugar, which has a hydroxyl group in the 2' position, whereas DNA has deoxyribose, which has a hydrogen in that 2' position. RNA has U, whereas DNA has T. And RNA is single-stranded, whereas DNA is double-stranded, comprised of 2 strands that run anti-parallel to one another. And this is just taking a look a little more closely at those DNA base pairs. So once again, they always occur between a purine, so adenine or guanine, and a pyrimidine, so shown here, thymine or cytosine. AT base pairs are connected by 2 hydrogen bonds. So you can see here we have a hydrogen bond between this partially positive hydrogen and this partially negative oxygen. And whereas guanine and cytosine are connected by 3 hydrogen bonds. So because these are connected by 3 hydrogen bonds, guanine-cytosine base pairs are actually bonded a little more tightly than adenine and thymine base pairs, because it requires more energy to break 3 hydrogen bonds than it does to break 2 hydrogen bonds. And if we look at that double helix in 3D, you can see here that the double helix actually has 2 grooves that go along its surface. First, you have a minor groove, which is more narrow. Then you have a major groove, which is wider. And because this groove is wider and exposes more of the bases within the groove, many proteins that modify the DNA will actually travel along this major groove. And once again, that phosphodiester backbone is found on the outside of DNA, whereas these bases, the nitrogenous bases, are stacked within the middle of the molecule.